At ASCO this year, we will be presenting an update on the IMGN632. This is a CD123 antibody-drug conjugate that has been developed for treatment of both acute myeloid leukemia as well as BPDCN, blastic plasmacytoid dendritic cell neoplasm. We presented some of the clinical updated data at the ASH 2019 meeting. This agent is initially being evaluated as a single agent in relapsed refractory AML, and the overall response rate that we see in this population is about 25 to 35% CR/CRI rate. So it does have efficacy, and it also had a favorable safety profile. We're not seeing any significant issues with very prolonged cytopenias. Importantly, we did see 2 cases of VOD, veno-occlusive disease of the liver. This was among about 80 patients treated. So there is a low but possible incidence. Both of the VOD cases occurred at high dose levels. We did a dose escalation from dose level 1 through 6, and the 2 VODs occurred, one at dose level 6, one at dose level 4. We're only going to be moving forward with dose levels 1, 2, and 3 because we were seeing similar efficacy between dose levels 2 and 3 and 4 through 6. So we felt that the biological effective dose would be safe, no VOD, relatively well tolerated, and giving us similar activity. So that is one of the posters that is still a trial ongoing in progress, single agent relapsed refractory AML. We're getting close to completing that trial. We did notice that in people who had non-therapy related AML, those patients had a higher response rate, 30-35% CR/CRI in salvage 1-2 situation. Now, in parallel, we have started a combination arm because, as most of the AML experts and treating physicians know, combination is the name of the game for acute myeloid leukemia therapy, and as single agents rarely, aside from molecular targeted drugs like IDH, FLT3, MLL, do we get great successes. So based on this, we are moving to a combination. This is a study I'm quite excited about. It has 4 different arms. There will be a combination arm of azacitidine with IMGN for salvage AML, first and second salvage initially, looking at starting at dose level 1 of the IMGN, going to dose level 2 and 3, and then selecting the biological effective dose, after which that study will go into an expansion where we will allow frontline patients to get AZA-IMGN as well as salvage. Now, of course, in the US, AZA-VEN is the standard, but the study does have a number of European centers where AZA-IMGN may be explored in a frontline setting just to get an idea of some safety and early efficacy. The second cohort is a Venetoclax IMGN combination. This was actually based on some very interesting and exciting preclinical data that Marina Konopleva's group presented as a poster at the ASH 2019, showing a high degree of synergy between the Venetoclax and the IMGN632 in PDX mouse models of different types of AML, as well as in multiple cell lines. So this combination will be in relapsed AML, again, looking at an escalation from IMGN dose level 1, 2, and 3, not going above 3, and will stay, of course, in salvage, not looking at any frontline, even in the expansion after the dosing has been determined. And the third one, which is probably the most exciting for us in the United States, will be a triplet of azacitidine, venetoclax, IMGN. This is currently ongoing. Again, for safety, will be evaluated first in relapsed refractory salvage 1 and 2 patients. AZA-VEN doses are standard based on the current label, and the IMGN will start at the lowest dose, dose level 1, then look at dose level 2 and 3. And then once we find a safe dose, and if we're seeing some encouraging efficacy, the plan is to evaluate this in the frontline setting. What we would want to see is improvement in depth of response, so more MRD negativity compared to AZA-VEN, which gives us about 45-50% MRD negativity. Can we improve that? And then, of course, duration of response and survival. It's going to be hard to beat the true CR/CRI rate per se in the frontline, which is 70 to 80%, but I think the depth and duration will be looked at. And so this cohort is enrolling nicely at this time. And the fourth one is looking at MRD eradication, of course, becoming one of the major areas of AML development. And this will be the single agent IMGN at the lower doses, dose level 1 or 2, which were quite safe in the phase 1. And these will allow patients MRD positive after any induction consolidation who are coming for MRD eradication approach. And I think within 10-15 patients, we should get an idea of whether we are seeing MRD log reductions or conversions to negative, and if that is being associated with a prolonged response duration. If it is, of course, this would be developed quickly in the future. So All these 4 arms are open and enrolling, a kind of exciting study to look at all these combinations in AML. And we hope to have data either by ASH or early next year to present. Thank you.